Paraphrasing Ralph Waldo Emerson, I shall try to show in this lecture how I've gone where there was no path and left a trail. Now, in trying to apply computational methods to complex systems, the essential idea has been to introduce classical concepts wherever possible to replace the much more complex quantum mechanics. And if we look at something that Dirac wrote in 1929, which many of you are familiar with, he said, the underlying physical laws necessary for the mathematical theory of a large part of physics and the whole of chemistry are thus completely known, and the difficulty is only that the exact application of these laws leads to equations that are much too complicated to be soluble. Many people know this, but they may not be as familiar that this was only the first part of a paragraph which went on. It therefore becomes desirable that approximate practical methods of applying quantum mechanics should be developed, which can lead to explanation of the main features of complex atomic systems without too much computation. This could be regarded as a leitmotif of this year's prize in chemistry, but actually what Dirac was thinking of was not classical mechanics, but simplifying quantum mechanics. Now, in trying to study more complex systems, we really have two aspects that we have to consider: the potential surface on which the atoms move and the laws of motion that govern the atoms, and both of these we would like to treat with classical methods where possible. Now, the Nobel Prize focused on the potential surface, and I will, partly to save time, I won't read everything that is said here, but basically the point is that both Michael Levitt and Ari Varshall come from the Lifson group, which was one of the pioneers in this area, and they will probably tell you about the potential surface simplifications. What I would like to focus on is the laws of motion for the atoms. And repeating in some sense, although the laws governing the motions of atoms are quantum mechanical, the essential realization that made possible the treatment of the dynamics of complex systems like proteins was that classical mechanical description was adequate in most cases. And from my own perspective, this realization was derived from simulations that we did in the 60s of a very simple reaction, H plus H2 exchanging atoms. And just to make this clearer, we have three atoms, a molecule AB and an atom C. The atom C collides with the molecule and leaves a new molecule, BC, and an atom A. Now, in order to do these calculations, we of course needed a potential surface, as I've already mentioned. And what we used here was a semi-empirical valence bond surface, as it's called. I was a student of Linus Pauling, and Linus Pauling believed that valence bond theory could solve everything. And so Dick Porter and I developed what has now become known as the PK surface, which is quite accurate. It's not perfect, as has been shown by later calculations, but I show down below here the energy as a function of the reaction coordinate, and the essential point is that there's a high barrier to go over for the reaction to take place. Now, I've shown the linear collision, which happens to be the lowest energy collision, but of course the actual reaction takes place in 3 dimensions. And let me just show you here, we are using classical mechanics to describe the reaction, and since there are only 3 atoms present, we can describe the reaction in terms of 3 coordinates, the 3 distances between the atoms, which is shown here on the left. And it's the distances between the atoms as a function of time in femtoseconds, which turns out to be the reasonable time scale. And as you see in yellow is illustrated the region where there's strong interactions. This is a reactive collision. And so, as you see, the molecule AB comes in, and the atom C comes in here. And at the end, you have a new molecule formed, the BC molecule, and the atom A goes out. Now, shortly after this, in 1967, with some primitive ray graphics as was available at Harvard, we made this film which shows the reaction. And you see the molecule vibrating, the atom coming in. Whoops, I guess, sorry. Hopefully this will remind you of what I've said already. Here again, you have the atom, the molecule vibrating, the atom coming in. It interacts with one of the atoms and goes out again with vibrational excitation. In the next slide, we have a non-reactive collision, again in 3 dimensions. Again, the interaction time is short on the femtosecond time scale. And you may ask your question, why are reactions even at high temperatures, like 1,000 degrees, which is the reasonable temperature for this reaction, why don't they occur on the femtosecond time scale? And the obvious answer is the reaction rate is determined by a large number of collisions. Most of these collisions don't have enough energy to get over the barrier. And so even though an individual event is very fast, the overall rate constant is many orders of magnitude slower. Let me show you here this non-reactive collision, which is, see there's again this interaction region, and the interaction is such that the molecule is highly rotationally excited. Now I want to mention one thing, a problem that arose when we tried to show this. Obviously what we're trying to do here is to make the atoms go larger as they come forward and become smaller as they go backward. And the question was, how do you introduce this perspective? Now this has nothing to do with the Nobel Prize, but it's something that I'm proud of, is to find a good law for showing perspective. And what I did was to visit Venice, which I actually did many times, and looked at the paintings of Canaletto and the actual distances and how he presented perspective. And it turned out that he used an exponential law, e to the alpha r. I don't remember the alpha. And so if I had published this, there are many things that I did which were not published, but maybe there would be a Karplus law in R theory as well as the Karplus equation in nuclear magnetic resonance. Now, one of the things that was essential in my development of these applications was to make sure that this would look like a very reasonable description of the H plus H2 reaction was actually the correct description. Obviously, to do that, you had to use quantum mechanics. And it was about 10 years later that a good friend of mine, Aaron Cooperman's group, and Wyatt's group were able to do an accurate quantum mechanical calculation. Since we had used an approximate potential to describe it, to test the methodology, this was not to test whether it agreed with nature, but to test our methodology, they used exactly the same potential as we had used, and they found that the classical results were as accurate as the quantum mechanical results that they obtained with much, much more work. And so the conclusion from this is that if a light atom like hydrogen atom, where you would expect the largest quantum effects could be described classically in most cases, not in all, there's tunneling and so on. that for the larger atoms, carbon, nitrogen, and oxygen, the heavier atoms of which biomolecules are composed, classical mechanics should be valid. But before turning to the larger biomolecules, let me go back a little bit to one of the items that was actually mentioned in the Nobel Award. I had been very interested in vision at Harvard, and in vision, the essential element is the photoisomerization of the chromophore retinal, which initially is 11-12 cis, cis meaning that around this double bond, the organization is as indicated, and when it photoisomerizes, it goes to 11-12 trans. And this photoisomerization is, if you like, gives the kick, which starts the visual cycle. Now, one of the things that we wondered about in the 11 cis and the chromophore ready to be photoisomerized, you have the possibility that around the 12-13 bond, it can be cis, it's called S-cis because it's a single bond, or it can be S-trans. And if you just look at the two, you would say, well, you don't need any high-level calculations, that obviously this one is going to be lower energy than this. Now, it actually turns out when you do the calculations, because the single bonds can twist very easily, these are very close in energy. Our calculations showed that this was lowest in energy, and this had various implications for the visual cycle, so we submitted it to Nature, and of course immediately got back the answer, well, there are no experiments that show that it's correct, so how can we publish it? Not realizing, and this is of course still true, that once there is an experimental result, then you're no longer making a prediction. And so I was sufficiently annoyed about that, so I wrote John Maddox, who was the editor of Nature, and said, look, you should really publish this, it's important. And he, after maybe an hour of discussion, said, okay, we'll publish it, we'll see what happens. They published our prediction that this was the lowest energy chromophore, and fortunately for him and for us, about 6 months later, an X-ray structure by Carl and Carl was published, which confirmed our result. But, so let's go on now and talk about the isomerization, which Marshall, when he was still a postdoc with me, we developed the method of using classical trajectories of the type for H plus H2 with a surface crossing model to be able to treat the photoisomerization. Here's a very simple case, 2-butene, where you have the two methyl groups on the same side of the double bond. After the light strikes, you get the trans-butene with the two methyl groups on opposite sides. Now, one thing that Marshall did very shortly after he left was to have the idea that in the protein might restrict the system from isomerizing if the two ends had to move. So he did a calculation which is illustrated here. He basically fixed the two end groups, threw away the rest of the protein, and then said, how can this isomerize? And he came up with this very clever bicycle pedal model, in which case you could get isomerization without moving the end groups. More work has been done on this since then, and it shows that the isomerization is similar to what he suggested, but it's somewhat more complicated. And when you actually include the protein, the rhodopsin, the protein relaxation plays a significant role. At the same time, actually, in 1976, we did the first calculation applying the trajectory methods to a protein. It's a small protein, has 58 amino acids and on the order of 500 atoms. We use the potential, was slightly modified from the Schrager and Lifson group potential that was developed by Bruce Gellin. And we did essentially exactly the same thing as if we had done for the 3-atom H plus H2 system, except we now had 500 atoms. But still classical equations of motion are applicable, and so it was a very natural generalization. Now I should mention that other people, I guess, hadn't worried as much as I did about whether the classical mechanics was applicable, but a pioneer in molecular dynamics, Anis Rahman, at about the time of the late 60s, had done already some simulations of liquid argon. And so this simulation of this very small, uninteresting protein, we still don't know exactly what its function is, gave results which the graphics weren't good enough so that we could actually illustrate what happened with the graphic systems. And so Bruce Gellin made this drawing of the inhibitor at the initial stage and after 3.2 picoseconds. And if you look, you can see that the structure is essentially the same, but everywhere the residues, represented by a single point, have moved. Now, to bring us up to date, Viktor Ovchinnikov, one of the members of my group now, took essentially the same system with a slightly different potential, and we see that there are these local fluctuations everywhere which occur in a system like this. And in an oral history that Andy McCammon had recorded somewhat later, he had made this wonderful statement, which was, there was a sense, even at the time, of something truly historic going on, of getting these first glimpses of how an enzyme molecule, for example, might undergo internal motions that allow it to function as a biological catalyst. Obviously, what we know nowadays, where there are thousands of simulations of this type being done, that what we felt at that time was really the beginning of a new era in the understanding of biological systems. Now, of course, as computers improve, one could improve the simulations, not only by improving the potentials, but also by doing longer simulations. And it was in '88, about 12 years later, that Levitt and his student Sharon did a 20 times longer simulation. And what was very important, they did it in a box of water molecules. Obviously, proteins, well, they don't exist really in water. It's a rather complicated cellular fluid. But nevertheless, this is a better representation than the vacuum one that we had to use in the early work. And they, I would say, qualitatively, they confirmed what we had found. They found that the motions of the residues were slightly smaller, and that because of the water friction, they were slightly slower. Then, much more recently, with specially designed computers, Shaw and his co-workers have done a simulation that's 1 millisecond in length. And to us, I mean, not very much interesting happens. You get somewhat more global motions in the protein. But to us, what the most important thing was the following. We always were scared, I suppose is the correct word, that these potentials which worked for 10 picoseconds or 100 picoseconds, if you simulated the system for a millisecond, it would just fall apart. And the important result, as I say, from my own point of view, was that the protein was perfectly happy, and even with these crude classical potentials, the system worked as it should. I mean, it's a very difficult thing in this area to be able to check that you are correct, because there are no detailed experimental data. I mean, we do need experiments still, in spite of what the Nobel citation says, particularly to be sure that what we're doing is meaningful. I won't have time to talk about protein folding, which is another very interesting area nowadays. But then we're going to go on to the next slide, which reminds me that, these motions are present in proteins at room temperature. And like everything else in nature, nature will have made use of them by evolution. And this is, again, this very well-known statement, everything that living things do can be understood in terms of the jigglings and wigglings of atoms. But you might be interested that 2,000 years before, a Greek poet, Titus Lucretius, made the following statement: the atoms are internal and always moving. Everything comes into existence simply because of the random movement of atoms, which, given enough time, will form and reform, constantly experimenting with different configurations of matter, from which we will eventually merge everything we know. Now, what exactly he meant by that, I have no idea, but it really is an amazing statement. And he never wrote anything else, but there it is. So the final part of the talk, really, I would like to indicate how nature has developed the structure of proteins to be able to use the motions to put them to work. And in a qualitative sense, there's sort of two parts to this. I mean, evolution determines the protein structure, and it's a structure in many cases, not in all cases, where the protein is made up of partly rigid units and then hinges which allow the rigid units to move one relative to the other. And then there's some sort of signal, usually the binding of a ligand, which changes the equilibrium between one structure and another. And let me show you a particular enzyme which has been studied by many people. But what this enzyme does, here you have adenosine diphosphate. Two of these come together in the enzyme, and one of the phosphates is transferred to the other one. So you have adenosine triphosphate and monophosphate. And like many enzymes, this one has an open structure which permits the substrate to come in and, of course, the product to go out, and the closed structure where you have a sort of reaction chamber which is isolated from the surroundings where the reaction can take place. And this is, again, a cartoon of what happens. As the two substrates come in, you have the open structure. It closes onto the substrates, and then over here is shown that the reaction takes place and the products go out. And it's an interesting point in this reaction and many others. The chemistry has been optimized so much that the chemistry is no longer the rate determining step. These are perfect enzymes in what Jeremy Knowles' way of describing things, that the rate determining step of the overall reaction is not the chemistry, but rather the opening of the system to let the products escape. Now let me finally turn to one particular system that's been of great interest to us. Molecular motors are, of course, the sine qua non example of how the jigglings and wigglings are put to work to do something that nature wants and needs. And we've studied several different motors, but I will talk just about one of them because, of course, the Physiology and Medicine Prize was given about the function of vesicles, which provide nutrients and other materials which are necessary for the function of the cell. Now, one point that hasn't been wasn't stressed in these lectures, not all vesicles need to be moved from one place to another. But kinesin, which was discovered in 1985, is very important in the sense that it's a molecule that does in fact do the transport of vesicles. The vesicles are up here. We know almost nothing about their structure or how they are attached, but it's a molecule, well, it's actually two molecules that have each one has this globular foot, as you will see, and then they're tied together by a strand to form a coiled coil which holds the vesicle. And what the physiology and medicine lectures were mainly about is how the vesicles open and close to give up their cargo at just the right time. This is concerned with how they move around. And again, this is a cartoon, and we'll just watch it. The microtubules down below, and they're the two globular feet. And here's a real-time photograph, enlarged about 100,000 times, where the kinesin molecules are actually moving on the microtubule. But I now want to focus more specifically on how kinesin functions. And if you look carefully at this, you can see that kinesin walks in the sense that we walk. It puts the left foot forward, and then it puts the right foot forward, and then the left foot forward. Now, so that it is walking in one sense, but of course, it's not surprising that these two molecules tied together don't walk really very well. And to me, what the way they're walking is much more like a person who has artificial legs, who sort of walks forward like this, but that a molecular system without all the things we have to make our feet work can do this is amazing to me. Now we'll look at a little more detail, and without going into all of it, we can see, here we'll follow this next power stroke. All the products of the reaction are important, but ATP coming in, which you just saw coming in, is the key to throwing forward the back foot to take another step. And here is now an X-ray structure of the molecule, which again has obviously the two globular feet connected by this beta strand to form a coiled coil here. And we did calculations which showed that just having this one beta strand would not be a system sufficiently rigid to be able to do this power stroke. And so the question was how it happened, and then we realized that there was another beta strand here, the N-terminal one, the initial one, which was disorganized in certain structures but could form a two-stranded beta sheet. We call this the cover strand, and the two-stranded beta sheet the cover neck bundle. And in simulations that we did, which are shown here, the work of Wang and others, we see the two globular feet and the disordered cover strand, all of this one sitting on the model of the microtubule. The ATP binds, and the simulations show that you form this two-stranded bundle, which looks very much like a spring. And if you look at the details, it's a high energy construct which the simulation suggests, in fact, acts like a string and throws forward the back foot to give you the beginning of the next step. Mutation experiments have been done to support this model, but we're still working on trying to show that it is really correct. So just make a few remarks about the kinesin motors. They're very important. I mean, if they don't work, mitosis is inhibited because kinesins carry things very backwards and forwards. Obviously, axonal transport, and kinesin was first discovered in the giant axon of the squid, so the vesicles can't get to where they're supposed to. And finally, it's sort of interest that viruses, who also are very clever, have learned that if they attach them where the vesicles are, the kinesin following the microtubule roads in the cell will get them across the cell in a few minutes instead of the 10 hours that it would take by diffusion in the complex cellular medium. So what does the future hold? Obviously, we all know that predictions are hard, but these are pretty conservative ones. And one thing that was already mentioned in the introduction and was really sort of my dream, if you like, when we started doing these simulations, is that not that they would take over from experiments, but rather that experimentalists would use them as a tool, just like X-ray or NNR or whatever, to study systems and get a better understanding than they could with either one type, experimental type, or theoretical alone. And that is certainly beginning now, and the hope is that the Nobel Prize, which says what we've done is something important, will aid in this. In terms of actual simulations, people are going toward more complicated systems, simulating viruses, ribosomes, cells. If I were 30 years younger, I would be simulating the brain, but I'm not. And so this future, we hope we'll see. Some of it is already in progress. But I, again, want to caution, as I do my students always, that what they're doing has limitations, just like do experiments, and they have to be careful, particularly when they find something interesting, that there isn't some error in what they have done. Finally, let me show you the people to whom this lecture is dedicated. These are the Karplusians, 244 people who have worked in my laboratory, if I can call it that, in Illinois, Columbia, Harvard, Paris, and Strasbourg. And it's to them that I dedicate this lecture, because without them, I wouldn't be here. And finally, let me mention one other person, my wife, Marcy, who is down here, who was willing to live with a person who spent all his time working, but even more than that, she was brave enough to be my laboratory administrator and made possible our movement from one part of the world to another. And in preparing for coming here, the complexity of everything that one has to do to be in the right place at the right time, I would have never kept up with it if she hadn't been there. Thank you very much.